Welcome back, friends. In this video tutorial we'll be talking about transfection, and this PowerPoint was taken from Brian Anderson. It's a beautiful PowerPoint. Thank him for designing this PowerPoint, and I've added some more slides and small editing purposes, all these things, background and all this stuff. Otherwise rest of them are from Brian Anderson. So let's talk about it. So transfection is a process of inserting DNA or genetic material. Especially when you talk about transfection, you talk about DNA, insertion of DNA inside a cell, right? Cell means definitely inside the nucleus of the cell, because in eukaryotes we cannot just inject it in the cytosol. We can inject nucleic acid in cytosol, but that should be RNA or siRNA or shRNA, like that, because siRNA mediated gene transcription and several processing are very important in new modern age techniques. But simply transfection means transfer of nucleic acid inside a cell or a nucleus. Inside the nucleus in case of plasmids and DNA molecule, in case of RNA like siRNA, mRNA and all these things, we insert it in the cytosol. So in this whole PowerPoint presentation we will see what is transfection, what is the difference between transfection and transformation, purpose of transfection, how it works, experimental methods and processes of that, and the advantages and disadvantages. So what is transfection? As I've told you, it's a method of transporting DNA, RNA and or virus macromolecules, various kind of macromolecules, into an eukaryotic cell by using chemical, lipid or physical based techniques. So these are the techniques we usually use, either chemical based method, lipid based, the liposome based methods, or micelle based methods, usually liposome based methods, and physical methods like gene gun or electroporation, like that. So if you look at different type of methods like calcium phosphate and DEAE buffer, this method is applicable for the DNA transfection. Liposome based is also important for the DNA transfection, and also for DNA transfection we have polyamine based transfection processes. But we have chemical, lipid, and so all of these things is lipo based and this is chemical based. But except for that we have physical based, which is gene gun, where directly gene is inserted inside the cell with a small particle added with the gene, and also the voltage mediated delivery. So here we can see the different types of transfection properties. For example, and these are the different commercial name of the kits where we don't want to talk about that, but simply the process of DNA transfection we can use the chemical method like calcium phosphate and DEAE based methods. It works best for many cell types, and it takes a long time, like 12 to 24 hours for this process to occur. Transfection, DNA transfection, again we can use only calcium phosphate. It can take kind of 3 hours and more than that, and we can use it for HEK293 cells. This is a type of cell in cell culture, in mammalian cell culture, HEK293 cell, and in this kind of cell we use calcium phosphate mediated transfection. Takes 3 hours plus all this. But liposome based delivery works for many cell types, but it requires small amount of DNA. We don't need a huge amount, very small amount, tiny amount of DNA can be inserted, and sometimes it should be inserted directly in the nucleus. It is gentle on the cells, and also room temperature storage is okay, and it takes kind of 4 to 6 hours for this process to work. And we can add for polyamine based transfection, right? This is for hard to transfect, all those cells which are hard to transfect, like as a cell line passes by, the primary cells are not that good for uptaking any particle from outside. So for those cells we have polyamine based, right? And also this is effective in presence of serum, right? And it takes kind of 3 hours, right? And it is not that much cytotoxic as the lipids or liposome mediated process. siRNA, if you want to do the siRNA transfection, we can use the proprietary formulation where this is completely optimized for siRNA delivery. It's knocked down the target gene even at low siRNA concentration, because most of the cases we do this siRNA transfection to knock down functionality of a particular gene, to block the synthesis of certain proteins inside the cell by inserting siRNA from outside, and this can take a long time, like 20 to 72 hours. 1 to 3 days, huge duration is required. If you look at lipid formulation, this works for many cell types, and also it includes control proteins. It's for the protein, peptide or other macromolecule delivery, except for then RNA and DNA. So we use all those cases. It takes kind of 4 hours for that process. Or we can go for viral packaging, where we need to insert a virus inside the cell, and we use a calcium phosphate for this process to occur, time 3 to 4 hours. So these are the different techniques that we can use. But all these techniques we have seen, we have talked about liposome mediated, viral, as well as the chemical mediated processes. We haven't talked about the physical processes here. We'll see that later. So the purposes of transfection is to study the gene function. How? Because let's say we add a particular gene from outside. So what we can do, we can interfere with the expression of certain genes inside the cell. So we can exchange those things. We can block the expression, we can turn on the expression, and all these things we can operate from outside, then inside the cell. And obviously keeping the cell alive, that's a hugely important thing. And we can also use the expression of proteins, because obviously it's all a matter of expression. That's why we are doing it. Expression of genes can be turned on or off, and all these things are related with ultimately the protein production. So obviously the expression of protein we can study. Or we can transfer the DNA into embryonic stem cells, and that can help the embryonic stem cells to grow and develop to finally give us the fully functional individual in this case. So if we look at the process of how this works, we can have the chemical and lipid methods. So you can see the chemical method is calcium phosphate mediated method. Now remember, why calcium phosphate? Because calcium is positively charged ion, and calcium can bind with the negatively charged backbone of DNA, right? So you can see the negatively charged backbone, phosphate backbone of DNA. Calcium can bind with them, and then finally it can drag those calcium beads, it's like a bead structures of calcium phosphate added, and then this calcium phosphate take those DNA segments and precipitate and settle on the cell. And as it settles on the cell, then cells start to take all those DNA slowly inside by cellular mechanisms, which we are not going to talk much about it. But it's kind of similar with transformation in bacteria, how they take the DNA segment after precipitation of the DNA with calcium phosphate. The process kind of resembles like that. In this case, but in this case it does not separate the DNA strands. It takes the DNA strands intact inside the cell. On the other hand, if we use the polymer method, so we produce the polymer of, it's kind of liposome. Liposome means obviously it's made up with phospholipids, where we have surrounding the outside regions, which is in contact with water and water content, are hydrophilic. In the inner core region is also surrounding regions of hydrophilic, and so as inside is hydrophilic as well as outside, it can contact with water and all the solutes there, as well as inside is hydrophilic, so hydrophilic molecules can stay inside and then we can use it. So you can see here we have the negatively charged DNA, and we have a positively charged ribosome which is outside the positive charge, and you can place this DNA inside this liposome. Once the DNA is inside the liposome, we call it a lipoplex. So now, as the DNA is it's kind of vesicle like structure, it's a delivery vesicle that we can form outside the cell, and then we can place it so that this lipid structures, this liposome, is actually made up with phospholipids resembling the phospholipids of cell membrane. So obviously if we place them, and cell membrane is negatively charged, remember, outside, so the negative membrane will interact with the positive charged lipoplex, and now this lipoplex content will merge with the membrane, and as a result all this thing can insert, the DNA will be inserted inside. That thing is possible. Or in other cases, what happens, it will be engulfed by endocytosis machinery. Endocytosis is done, and then what we can see, inside the endosome we have the lipoplex, and in the lipoplex, inside the lipoplex we have those DNA contents inside. Now those endosome will now fuse with lysosome to degrade those DNA. That can happen, the DNA fragmentation. We sometimes require this DNA fragmentation and fragmented portion of the DNA to be inserted, or sometimes we don't want that. So when you don't want that, we allow the environment of the cell in such a way so that the liposome will not fuse with the lysosome. So what we want here is that this lipoplex, as you can see, the lipoplex and the endosome, they are kind of mixed, As they are kind of mixed with one another, this DNA becomes free and the DNA will be inserted inside the nucleus through the nuclear pore. On the other hand, if we look at the physical methods, there are plenty of physical methods out there, like the electroporation, where we use a high voltage to deliver the nucleic acid through the pores that we form. We apply high voltage in the cell, it creates a pore, but the pore will remain for a very fraction of second, then again that portion will be healed. Actually, the pore forms due to the separation of all those lipids from a particular area, then again they will come back, but during that time, when the pore forms, the DNA enters inside the cell, and then again those lipids will fill this gap and the DNA will be inside. Or we can use the micro-injection process, where you can see a very tiny needle. If you look at this picture, you can see this is the end of this needle, very tiny needle help to inject the DNA inside the nucleus directly. Usually we use this direct micro-injection properties and things in case of delivery of gene inside the embryo, right, during the development. Or if you look at the chemical methods of how we transfer things, this is kind of complicated methods. The protocol is huge, but for your understanding I'm going to talk about very little of this. So what we have two different solutions. We have solution A, which contains plasmid DNA, that means the DNA we want to insert inside the nucleus, along with the calcium solution, because calcium will coat all those outside surrounding region of the plasmid, because plasmid backbone is negative, calcium is positively charged. Now solution B has 2x-HBS. Now HBS is Hepis buffered saline. So this is buffered saline and now plasmid DNA with calcium solutions. Two solutions, solution A and solution B, are ready. Now those solutions will be mixed together. So if you add solution A and solution B and then we vortex those things, during the vortexing, what will happen? After this process we incubate for 20 minutes, and what we do then, apply the transfection solution to subconfident cell culture. Then once this process is stayed there for 20 minutes or so at a particular temperature, we take that mixture and add it to the cell where we need to insert those DNA. Then what will happen, then we again incubate it for 2 to 12 hours, and then after the 2 to 12 hours these transfection solutions will completely go inside, and then finally the DNA will be inside the nucleus and at the destination. So once that process is done, what we do, we assay for whether the gene is inserted inside the cell or not. We check that, and once doing that, then we take those cells which are getting our DNA, which are perfectly transformed, we take those cells and we transfer those cells to actually stable transformants, instead of taking because the solution we put them at the first, that is the solution here, but that is not the actual nutrient solution for cell line growth. So then we take those actual transfected cell and place them into the proper solution for long-term growth of those cells. So what are the advantages and disadvantages of transfection? Advantages is that it provides the ability to transfer negatively charged DNA into a negatively charged membrane, through the negatively charged membrane, and that is possible either using a chemical method or using the liposome mediated delivery. And liposome mediated transport of DNA has high efficiency, more than the chemical mediated delivery, but in this case it's slightly more expensive, and also this liposome mediated process kind of toxic in certain cases towards the cell. It is very good for long-term studies, right? And if you look at the disadvantages, the chemical reagents that we use are not useful for the long-term studies, right? It can damage the cell. And although the transfection efficiency depends on the cell health which is being transfected, the DNA quality and DNA quantity, this is the confluence of 40 to 80% is required, right? Then only those cells can be transferred to the next time for the growth. And direct microinjection and all those biolistic particle delivery, I've not talked about this. Biolistic particle delivery is a delivery using gene gun, where you have a gun like structure with gold particle in it, which will fire the gold particle will be wrapped by the DNA, and then we fire that particle, go inside, penetrating the cell membrane, go inside the cell along with that particle. So as it uses gold particles and all this, in this case that becomes expensive, right? And it's also difficult methods, require trained personnel to handle all this stuff. So these are the disadvantages, but there are most of the advantages, and it opens up a whole new arena of mammalian cell culture to transfect them with all these different genetic material to finally see the expression of genes, to control the expression of the genes from outside, to see turn off the certain expression using siRNA mediated transfer. So that is very important, and I hope that helpful. If you like the video, hit the like button, share this video with your friends in all the social networking site, as well as subscribe to my channel to get more videos like this. Help us do more videos for you. Thank you.